Okay. So the topic is the transcription in the prokaryotes and the transcription in the eukaryotes. Okay. So first you go for the transcription process in the prokaryotes, then you go for the transcription process in the eukaryotes. So before start a particular topic, first of all we have to know that what is a transcription process. What do you mean by a transcription process? Okay. So transcription process is nothing but, transcription means what? The formation of RNA from the DNA. Okay. The formation of RNA strand from the DNA is called as a transcription process. Now see, we all know different types of RNAs are there. Okay, it may be the rRNA, it may be the tRNA, it may be the mRNA. Okay, and all the different types of RNA, just like you can say miRNA, siRNA, hnRNA, different types of RNAs are there. Okay, so all the RNAs are transcribed from the DNA. Okay, and the process is called as a transcription process. Okay, so formation of RNA from the DNA is called as a transcription process. Okay, now see, so during the transcription process we all know DNA is found inside the nucleus. Okay, so inside the nucleus DNA is present. So transcription process always occur inside the nucleus because DNA is present inside the nucleus and RNAs are produced from the DNA. That means the process of transcription occurring inside the nucleus. Okay, and after the transcription the next process is called translation. Now what is translation? From the RNA they can produce the protein molecule. Okay, so RNA molecule can be converted to the protein. Okay, and this is occurring in the cytoplasm, not inside the nucleus. Why? Because RNA molecules, they are getting inside another cell organelle which is called as the ribosomes, because ribosomes are found in the cytoplasm, they are not found in the nucleus. That means the protein translation process is occurring in the cytoplasm, not in the nucleus. But the transcription process, that means the process of formation of RNA from the DNA, this is occurring inside the nucleus. Okay, so this is same in terms of the transcription process in the prokaryotes as well as transcription process in the eukaryotes. In both the process it is same, but somewhat some type of differences may be there. So in this chapter we discuss about what are the difference between transcription process in the prokaryotes and what is the difference between transcription process in the eukaryotes. Okay, now going for the presentation. Okay, so first topic is the transcription process occurring in the prokaryotes. Okay, the detail of transcription process occurring in the prokaryotes. Okay, so before going to start this process, first of all we have to know that which enzyme is responsible for the transcription process in the prokaryotes. Okay, and the enzyme which is occurring in the DNA of the prokaryotes and converted from the DNA into RNA strands, the enzyme is known as this one, DNA dependent RNA polymerase. Okay, so this is the particular enzyme. So this is the particular enzyme called as the DNA dependent RNA polymerase. Okay, just to remember this term, DNA dependent RNA polymerase enzyme. That means this RNA polymerase enzyme is very important and they depend on the DNA to synthesize the RNA molecule. Okay, now see, so this is the structure, this is the basic structure of the this one, the RNA, sorry, DNA dependent RNA polymerase. So in the DNA dependent RNA polymerase, the enzyme having different types of subunits are there. So they are having different polypeptide chains. Okay, so this is one alpha chain is there, another alpha chain is there. So that means they having the two alpha chain. Okay, then one beta chain is there and one is beta dash chain is there. Okay, so two alpha chain, one beta and one beta dash, then one omega chain is there, omega. Okay, so these 5 polypeptide chains are called as the core enzyme. This is called as the core enzyme. Okay, so this is the core enzyme of the RNA polymerase. Then another polypeptide chain is there which is called as a sigma. You can say sigma. Okay, this is another core enzyme. when the sigma molecule bind with this particular coenzyme, then they produce a new type of enzyme. This is a new type of enzyme, and this enzyme is called as a, you can see, holo enzyme. This is called the holo enzyme. That means this is the important enzyme which is called as a DNA dependent RNA polymerase, and this enzyme can transcribe the DNA into the RNA. Okay, and this is called a holo enzyme. So holo enzyme having the 6 different polypeptide chains are there. Okay, so 2 alpha, 1 beta, 1 beta dash, 1 omega, and 1 sigma. Okay, and this is the holo enzyme. So if the sigma factor is not binded to this core enzyme, then the RNA polymerase is functionless. RNA polymerase cannot transcribe DNA into the RNA if there is no sigma factor. Okay, so sigma factor is basically it is not attached to the RNA polymerase. It is present in the other parts of the nucleus. Okay, nucleus thakibo, but it's present in the other parts. So when the sigma factor comes and bind with the core enzyme, then they produce one type of enzyme called as a holo enzyme. Now this holo enzyme can transcribe DNA into the RNA. Okay, so this is the important holo enzyme called as DNA dependent RNA polymerase. Okay, and this is the structure of the holo enzyme. Okay, now we need to know about the process, the transcription process occurring in the prokaryotes. Okay, so it basically involves 4 different steps. Okay, 4 different steps are there which is occurring in the transcription process of the prokaryotes. Okay, the first point is called as the binding of RNA polymerase to the DNA. Okay, so I already said what is the structure of the RNA polymerase. Now this RNA polymerase have to bind to the DNA so that it can transcribe into the RNA. Okay, so question is that where the RNA polymerase can bind in the DNA. So there is maybe a specific region may be there. So in the specific region of the DNA, RNA polymerase can bind. Okay, so you can see number 1 point, this one, binding occurs at a particular site of the DNA called as promoter. Remember, the term is called promoter. Okay, so in the DNA there is a particular area is there. Okay, the area is called as a promoter. So in the promoter, the RNA polymerase can bind. Okay, and the region of the promoter is what? It is specific, 20 to 200 bases. Okay, the region is having 20 to 200 bases are there in the promoter region. Okay, in the promoter region from 20 to 200 bases are there. Okay, this is a specific region where RNA polymerase can bind, and this specific region is called as a promoter region. Remember. Then see, 2 specific promoter sites found in the organisms. Okay, in a region of 5 to 10 bases preceding the coding region in a specific sequence of 7 bases, this one with minor variation. Okay, now see, so in a promoter region there is a 5 to 10 base pairs are there. So total promoter region is what? From 20 bases up to the 200 bases, the total promoter region. So in this total 20 to the 200 base pairs, only 5 to 10 base pairs are there, which is very important. Among this 20 to 200, 5 to 10 bases are very important in the promoter region. Okay, approximately it is a 7 bases in between 5 to 10, but approximately is the 7 bases, which is very important for the DNA polymerase. And these 7 bases sequences are this one. You can see TA, TAA, TG. 1, 2, 3, 4, 5, 6, 7. These are 7 bases. Okay, and this sequence is very important for the RNA polymerase to bind. Okay, and this sequence is called consensus sequence. You can see this is called as a consensus sequence. It's a very important term, consensus sequence. Okay, so TA, TAA, TA, TAA, TG. So this is the sequence which is called consensus sequence, which is a 7 base pair sequences found in the promoter region. Okay, the promoter region is very large in structure. From 20 to 200 bases are there. In between large promoter region, only 7 base area is there, and this 7 base area is called consensus sequence, which is called as a TA, TAA, TG region. Okay. So why it is called as a consensus sequence? What is the meaning of consensus sequence? Consensus sequence means there is a little variation in this particular sequence. A very little variation occurs in the particular sequence. That means for every organism, this consensus sequence is almost similar for every organism. It may be bacteria, it may be virus, it may be some other mammals, it may be some amphibia, it may be reptiles, all having the similar type of sequence, which is called TA, TAA, TG. Okay, if the variation may be there, the variation is very minute, very little variation is there. That's why this sequence is called as a consensus sequence. Okay, now see, in the E. coli, this consensus sequence is called as the, this is the term, this is called as a Pribnow box. Pribnow box, remember, this is called as a Pribnow box. So TA, TAA, TG, this particular sequence of 7 base pair found in the E. coli is called as Pribnow box. Okay, this region is called Pribnow box in the E. coli. Okay, now this a little variation occurs in the eukaryotes. Okay, so in case of the eukaryotes, this sequence is somewhat little variation occurs. After the variation, they forming this structure. You can see TA, TAAA, T. So this is the sequence of 7 base pair, which is found in the eukaryotes. Okay, so somewhat little variation is there, but basically TA, TAA, this is almost similar. This sequence is almost similar. Only the variation is the TG. TG is varies into the AT region in case of the eukaryotes. Okay, and this sequence, which is found in the eukaryotes, it is called as the this one, Hogness box. Remember, Hogness box. Hogness box is the region which is found in the eukaryotes, and the Pribnow box is found in the prokaryotes. And little variation is there in between the Hogness box and the Pribnow box. And this 7 base sequence is very important for the transcription process in the prokaryotes as well as eukaryotes. Just try to remember these 2 boxes, T, A, TAA, TG in case of prokaryotes called as the Pribnow box, and then TA, TAAA, T, which is called as the Hogness box in case of the eukaryotes. Okay, and both are very important for the transcription process of the prokaryotes as well as the eukaryotes. Okay, and both these boxes are found in a region of promoter region, and promoter region is very bigger size region. This is constitute 20 to 200 bases. Among this promoter region, only 7 bases are there, which is very important, called as Pribnow box or maybe called as a Hogness box. Okay, then see, the binding of RNA polymerase to the DNA, it is continued. You can see this region is known as Tata box. So what is Tata box? You can see the previous region. You can see TA, TA, this is similar. This is the region TA, TA, and over here also this region is similar. So in case of the prokaryotes and in case of the eukaryotes, first 4 bases is similar, TA, TA. That's why this box may be also known as Tata box, TA, TA box. Tata box boli jetia kobo, when someone asking you about the Tata box, that they can say about the Tata box may be for prokaryotes or Tata box may be for the eukaryotes. But when they are specific, when they are saying about Pribnow box, then Pribnow box only for the prokaryotes and Hogness box only for the eukaryotes. But when they say Tata box is common for prokaryotes as well as the eukaryotes. Okay, then see the region known as Tata box. In this region where double helix open to form open promoter complex. Okay, that means from the Tata box in this region, from the Tata box region, the double helix is unwinding. The double helix molecule is the unwinding, and they can separate into two strands from the Tata box itself. That means the initiation of the replication, the initiation of the transcription is start from this particular point. The initiation of the transcription is start from the Tata box point. That means the open promoter complex is formed from the Tata box region. Okay. Then see, the center of Pribnow box lies usually 10 base pair upstream. Okay, 10 base pair upstream, and it contains T, which is called as a -10 sequence, which is recognized by the RNA polymerase. Okay, now see. So there is a direction. There is a direction of the RNA synthesis from the DNA. There is a direction. Now, what is the direction? That means the DNA having the two strands are there. Okay, in the DNA, two different strands are there. From the two strands, only one strand can transcribe into the RNA, not both the strands. Just to remember, from both the DNA strand, only one DNA strand can transcribe. Okay, it may be the upper strand or maybe the lower strand. Jikono eta strand ki hobo transcription hobo. So during this transcription process, okay, so only one strand can transcribe into the RNA. We all know. Now see. Now question is that which strand undergoes RNA transcription? Which strand undergoes the RNA transcription? So just to remember, there is a direction. The direction is the 5' to the 3' direction. Okay, 5' to the 3' direction, the RNA can be synthesized. Okay, now see. Suppose this is one strand and this is another strand. Okay, suppose I have written over here, suppose this is the 3' end, suppose over here this is the 5' end. This is the 5' end. Suppose this is 5' and suppose this is the 3'. Okay, so both the strands are suppose separated. Both are separated. Separate hoye gol. Both the strands are separated. Okay. Now question is that in which direction the RNA polymerase move and they transcribe the RNA. From which direction? Okay. The direction is 5' to the 3'. That means over here there is a 3' and there is a 5'. So the movement of the RNA polymerase is opposite. That means suppose RNA polymerase is binding over here. Suppose this is the RNA polymerase. Then the RNA polymerase is moving in this direction. This direction. Why? Because this is 3', this is 5'. That means this direction is what? This direction is 5' to the 3'. Okay. So RNA is synthesized in this direction. That means this is the template strand. This is the template strand. The template strand direction is 3' to the 5'. The direction of the template strand is 3' to the 5'. Okay. That means this strand is transcribed. Okay. And this strand is not transcribed because in this strand the direction is 5' to 3', opposite. So RNA polymerase is moving from 5 to the 3' end. That means this strand is transcribed and this strand is not transcribed. Okay. So this is the direction of the RNA polymerase to synthesize the RNA. Okay. And this is the template strand and this is the non-template strand. Okay. Now see, over here it is written as the center of Pribnow box lies usually 10 base pair upstream. Okay. Now what is 10 base pair upstream? Okay. And what is -10 sequence? Okay. Now see. Suppose this is a normal DNA. This is a normal DNA. Okay. So in this DNA, suppose this is one region and this is another region. Okay, and this is another region. Now see, so this is the region of the DNA. This is another region of the DNA, and this is the start region. You can see start region. Now what is start? Start means the synthesis of the RNA start from this area. It started in this direction. It is started. Okay, in this direction. Okay, that means the RNA polymerase have to bind in the upstream area. So this area is called upstream area. Okay, so RNA polymerase is binding in this area, and they are moving this direction. This direction they are moving. Okay, now see, here it is written as upstream -10 sequence. Okay, suppose this is the +1. Suppose this region is called +1 sequence. +1 sequence. Then this is what? -10. This is -10, and suppose this is -35. Okay, -10 sequence and then -35 sequence, and this is +1 sequence. That means the synthesis occurs this direction. Now see, so RNA polymerase binding in this area, which is called -10. And what is -10? -10 having the Pribnow box. So TA box, the 7 base pair sequence is found in this area. A area to thakibo. So yat ki thakibo tomar T A then T G. Okay, so this sequence is found in the -10 region over here. Okay, so this is the Pribnow box which is found in the -10 region. What is called -10? Because the synthesis start from this one, +1. Okay, and this region, this region is called promoter region. The whole region is called promoter. This is called promoter. This is the promoter region. I already said promoter region is very big region. It having 20 to the 200 number of bases. So promoter region again extend up to this direction. Suppose this is the 200 base pair. Suppose it is the -200. Okay, so starting from here up to here, this the whole region is called promoter region. This is the whole promoter region up to the -200 base pair. Okay, so in this promoter region, two important regions are there. One is called -10, one is called -35. So in the -10, this Pribnow box is found, which is called TAA, TG. This is found. Okay, and in the -35, there is another region where the important molecule is there, which is binding in the -35 region. The important molecule is called sigma. So sigma factor is binding over here. The sigma factor. RNA polymerase having a sigma factor. So sigma factor is binding in this area, and this region is called -35. Okay, and when the sigma factor is binding, then RNA polymerase come and bind over the sigma factor, and they fill up the whole area. They fill up the whole area like that. They fill up the whole area. Okay, and so in the DNA, different regions are there. Remember, in the DNA, different regions are there. Suppose starting from this up to the 200. Okay, this is a specific region which is found in the DNA, and this specific region is called promoter region. Remember, the specific region which is found in the DNA, the length of the promoter region is from 20 base pair up to the 200 base pair. The length may be smaller, 20 base pair over it, but up to 200 base pair. Okay, because every DNA having different types of promoter region, but basically the length of the promoter is up to 200 base pair. Okay, now think about, this is a promoter which length is 200 base pair, suppose. Okay, so in this 200 base pair length of the promoter, two important sequences are there in the promoter. Okay, the first sequence is called -10 and second sequence is called -35. Okay, now what is -10 sequence? So in the -10 sequence, the Pribnow box is found. So Pribnow box is found in the -10 sequence. Okay, over here. So TG, this sequence is found in the -10 region. Over here, it is found. Okay, sir? And in the -35, another sequence is there, and this sequence is very important for the binding of the RNA polymerase, because in the -35 region, in this region, the sigma factor of the RNA polymerase bind. RNA polymerase having two important structure, remember, you can see. The sigma factor is there and the core enzyme is there. Both are separated. Sigma factor and the core enzyme both are separated. So first, this sigma factor is binding to the -35 region of the promoter. In the -35 region of the promoter, the sigma factor is binding. After the binding of the sigma factor, this particular another core enzyme, this is bounding over the sigma factor, and they form the holo enzyme. Okay, and when they form the holo enzyme, then they start the transcription process. Okay, remember, sigma factor is binding to the -35 region of the promoter. Okay, so over here the sigma factor is bind. So when the sigma factor is bind over the sigma factor, because the RNA polymerase is a very bigger structure enzyme, so this enzyme covers the whole area. They covers the whole area, -35 up to the -10 up to plus this one. The whole area is covered. A gote area to cover koribo. Okay, and they binding over the sigma factor, and then this particular enzyme now called as the holo enzyme, and this holo enzyme can start the transcription process in this direction. Okay, now understood? So this is the important binding features of the this one RNA polymerase, how they can bind. Okay, in the -35 region, it's a long region of -35 sequences. So in this -35 region, there is a small region called as a 9 BP consensus sequence. So in this -35, a small region of 9 base pair consensus sequence, okay, which is very important to binding of the RNA polymerase. You can see RNA polymerase, binding of the RNA polymerase. So RNA polymerase is actually binding the 9 BP region of the -35. That means what? In this 9 BP region of the -35, this sigma factor is binding. Okay, the sigma factor is binding in the 9 BP consensus sequence found in the -35 region. So 35 region is a very big region. 35 base pairs are there. In this 35 base pairs, only 9 base pairs are very important, which is called 9 base pair consensus sequence. And in this sequence, the sigma factor bind, and over the sigma factor, next core enzyme bind over there, and they becomes holo enzyme. And when they convert it to the holo enzyme start now synthesizing the RNA from this direction, Okay, now question is that why they moving this direction? Because this particular RNA polymerase, they always synthesize to the 5' to the 3' direction. 5' to the 3' direction. Now see, this is the concept. This is the strand which is having the 3' to the 5', and this is the strand 5' to the 3'. So synthesis always started 5 to 3. So this is the enzyme binding over here. When the enzyme binding over here, then this is the template strand. This is the template strand which is 3' to the 5', and synthesis started from 5 to 3. So this is the direction. This is the direction. Okay. Now you can ask me, why not this enzyme binding over here? Why they are binding on this direction only? Why they can bind over here also? Yes, it is possible. It can bind over here also. When the RNA polymerase binding over here, যদি ইয়াত bind হয়, then what happened? The template strand is now different. You can see. So if the RNA polymerase binding over here, this is 3' to the 5' area. Okay, and this is 5' to the 3' area. That means in this, when the RNA polymerase binding in this direction, then the template strand is this direction. They can synthesize, and template is this one now, because this is 5' to the 3' direction. Okay, now this is the template strand, and this is the non-template strand. Now understood? When the RNA polymerase binding this direction, then this is the direction of synthesis. Now this become template and these become non-template. And when the RNA polymerase is binding over here, then this become template and this become non-template. So based on the binding of the RNA polymerase to the DNA, then template may be different. If they are binding to the left side, then the template becomes the upper one, which is the 3' to the 5' direction. When the RNA polymerase is binding to the right side, then the template becomes the lower one, which is the 3' to the 5'. Okay, so this is the difference of binding region of the RNA polymerase to the DNA, and based on that they can select the template. Just to remember, the template have to be the direction of the 3' to the 5'. Template direction is the 3' to the 5', and the synthesis of the RNA strand is the 5' to the 3' direction, opposite direction. Template is 3 to 5 and the synthesis of the RNA is 5 to 3. Okay, so based on the binding of the RNA polymerase. Okay, so this is the binding region. So after the binding, then they start the initiation process. Now what is initiation? Okay, now see. Next process is the initiation of the transcription. How they initiate the transcription process. Okay, now see. RNA polymerase contain two nucleotide binding site called initiation and the elongation site. Okay, so in the RNA, in the DNA molecule, two important areas are there, which is called initiation site and there is a elongation site. Two important sites are there. One is called initiation site, one is called elongation site. Okay, now see. The initiation site bind only purine triphosphate, namely ATP, GTP. Usually ATP is the first nucleotide of growing RNA chain. Okay, now see. When the RNA polymerase binding to this particular DNA, okay, you can see this is the -35, the sigma factor is binding over here, and this is a Pribnow box, and this is a starting point. From here, the RNA polymerase started the synthesis this direction. Okay, now see. When the RNA polymerase is binding to the DNA, then what happens? Then the unwinding process occurs in the particular DNA. The DNA becomes unwinded. Both the hydrogen bonds are breaked and the DNA looks like this. Okay, and the RNA polymerase is moving like that. Suppose this is 3' to the 5' is the end. Then the synthesis of the RNA occur this direction. This is the synthesis of the RNA. Okay, and this is the non-template strand. Now see, when the RNA polymerase is binding over here, the first base, this is the first base, suppose this is the first base, starting of the first base. So this base which is produces in the RNA strand, this base have to be the ATP, base have to be the adenine. Adenine have to be the binded over here. Adenine is binded. So if the adenine is binded over here, the opposite molecule is have to be what? They have to be the T. Is a T molecule because T is binded to the A. Okay, so the first base which is found in the start region, the first base which is found in the start region always have to be the T. It always have to be the T molecule because this T is binding to the A. The A molecule is the ATP. ATP. This is called adenosine triphosphate. So adenosine triphosphate binding to the T molecule. Okay. The second strand may be G or maybe C, maybe suppose again G, maybe A, like that. But the first base always have to be T because the T is binding to the A. You can see the initiation always starting with the ATP molecule. ATP molecule always starting the initiation. If the ATP molecule always starting the initiation, that means the opposite strand found in the DNA always have to be the T. T hobo lagibo too because T is binding to the A. Okay, so this is the initiation of the transcription. Initiation always start with the ATP molecule bind to the T, so that T and A both are binding to each other. They becomes complementary to each other. T always bind with the A with double bond hydrogen bonding. Okay, so this is the initiation step. Then see, the first DNA base that is transcribed is usually the thymine. Is a thymine. You can see. So this is the first base, usually is the transcribe is a thymine molecule, because thymine molecule is binding to the A molecule. The first base which is binding in terms of the RNA always the T. Sorry, so it is always the A molecule. So this A molecule they have to be binding to the T like that. Okay. Then see, the elongation site is a field with the new base complementary to the DNA template, and both the bases are joined together and then first base is released. Okay, the most important point, second point, you can see the second base. Now first base is complementary T and A, then go for the second base. Suppose this is a G. If it is G, what about the this RNA? Jodi to G thake ki hobo yat C hobo. Now it becomes a C molecule. If it is G, it is a C. Then C is binding over the G, and this A and C both are bonding to each other. A molecule and the C molecule both are binding to each other. Okay, and then the synthesis of the RNA is continuous. Okay, first they synthesize A, then C. Suppose this is C, then they produce the G. Suppose this is G, then they produce the C. Suppose this is A, they produce the U, because it's the RNA. A hole U hibo, T nahe. A always binding to the U molecule, because this is the RNA strand, not a DNA strand. In the opposite of the A, it is the U. Okay, and this synthesis is going on like that. Okay, so first RNA always have to be the A. Second is depend on what is present in the DNA. In the DNA, if it is G, then the RNA is the C. If it is a C, then it is a G. If it is a G, then it is a C. If it is the A, the RNA is the U, like that. Now see, the most important concept, just try to remember this point. When the second base is binding over here, suppose the second base is G in the DNA, RNA have to be the C. Then A and C both are binding to each other. A and C both are bonding to each other. After the bonding of the A and C, what happened? The A molecule is released from the T. The hydrogen bond between the A and T, it is dissociated, and A molecule is released. Okay, RNA polymerase is moving forward and A is coming out like that. A is coming out like that. This is the A. A is coming out. A always bind to the T. We know. The second C is binding to the A, and they can form the bond between A and C. Okay. Now see, it may not be C. It may be Z. It may be U. It may be C. Why? It is depend on the template strand. Template 0 thakibo, opposite yat bind hobo. Template jodi G ase yat C bind hobo. Template jodi A ase yat U bind hobo, like that. Okay, but the first molecule always have to be the T. Yat hodai T thakibo lagibo, because T is binding to the A. Because the first RNA molecule always having the A. If it is A there, the DNA is always have to be the T. Okay, the second base, suppose over here G is there. Then the opposite RNA is the C. Then A and C both are binding to each other. Okay, so when the next concept is there, when the C is binding to the A, when the C molecule is binding to the A, immediately the A is detached from the T. The A molecule is detached from the T. The hydrogen bonding between the T and A, it is detached, and the A molecule is released. But they binding to the C. The binding between A and C is tightly binding, but the binding between A and T is loosely binding. It is detached. This binding is removed and A is released. A molecule is released like that. A molecule is released, but C molecule is attached. C molecule is attached over here. Okay. Then when the RNA polymer is moving forward, ako jeta move koribo ifale, suppose next molecule is C. Jodi next molecule is C hoy, yat ki bind hobo, G hobo bind hobo. Then G is binding. When the G molecule is binding, then immediately the C molecule is detached and C molecule is coming out like that. C molecule is coming out. Okay. Then again the RNA polymer is moving like this direction. Suppose next molecule is G. When the next molecule is G, it is binding with the C. Then this G molecule is detached and G is coming out like that. G is coming out. Then suppose next molecule is A, then it is binding with the U. then this C molecule is coming out like that. Okay, and this is coming out like that. The strain is coming out. Okay, so the strain is A, C, G, C, U. It is coming out like that. So RNA polymer is moving this direction, and the RNA which is synthesized, it is also detached and coming out like this direction. In the opposite direction it is coming out. Okay, so RNA polymerase is moving forward, and the backward direction, the RNA which is synthesized, it is also coming out like that. But the bonding between A and C is not detached. The bonding between G and C not detached. The bonding between G and C not detached. The bonding between C and U not detached. The bonding between A and U and A not detached, like that. The bonding is tightly bonded. But the hydrogen bond, this is a hydrogen bonding between the above the DNA, it is detached and it is coming out like that. Okay, so this is the main elongation process of the RNA. So when the RNA polymerase is moving forward one direction, they unwind the DNA. Every single bases are coming out. They binding to the complementary DNA. First they binding to the complementary DNA. Then when the RNA polymerase is moving forward, the bonding between the DNA template and the RNA is detached. The hydrogen bonding is detached and they are coming out. They are moving, and every movement of the one base pair is coming out like that. Okay, and finally when the RNA polymerase is reaching to the end point, suppose this is the end point of the DNA. So in the end point, when the RNA polymerase reaches, this RNA chain is detached from the RNA polymerase, and this RNA chain is coming out completely like that. This is the RNA chain. It is also detached from the RNA polymerase. Okay, and this is the newly synthesized RNA which is formed finally. The elongation process. Elongation process. Now what is elongation process? You can see this is the RNA polymerase enzyme. This is the RNA polymerase enzyme, the bigger enzyme. Okay, so inside the RNA polymerase enzyme, the unwinding process is occurring. The unwinding process is occurring. Okay, now see, here a question you discuss about the DNA polymerase 3. We will discuss about the DNA polymerase 1. Okay, so DNA polymerase 1 and DNA polymerase 3, they cannot unwind the DNA. Okay, DNA polymerase 1 and DNA polymerase 3, they cannot unwind the DNA, because for unwinding of the DNA, a separate enzyme is needed. The separate enzyme is called DNA helicase. So DNA helicase can unwind the DNA, but this is exception for the RNA polymerase, because RNA polymerase do not need the DNA helicase molecule. RNA polymerase itself unwind the DNA. RNA polymerase itself unwind the DNA, and also they can again rejoin the DNA. Unwinding process and the rejoining process both are occurring by the RNA polymerase. This is the strain, and again both the unwinding DNA again rewind. Again they joining the both the DNA. Okay, so you can see the direction. Okay, you can see the RNA polymerase is moving this direction. This is the unwinding process. Okay, during the unwinding process, this is the template strand is binding like that. This is a template strand. When the template strand is binding, then what happens? This is released. This strand is released. This strand is released like that, and the DNA polymerase is moving. Sorry, RNA polymerase is moving this direction. Okay, so when the RNA is moving this direction, what happens? This DNA molecule again rejoin. They are again rejoined. They again rejoin. Okay, so when the RNA polymerase is moving this direction, unwinding process is going on this direction. This direction unwinding process is going on. RNA molecule is released, and this side DNA again rejoin. Rejoining of DNA occurs over here, because RNA is moving from this point to this point is moving. If RNA polymerase is moving, suppose this direction. Suppose this area is free. Now this free area, the DNA again rejoin. Again they rejoin. Jodi T asile, T is binding to the A again. Jodi G asile, G is binding to the C again. Jodi yat C asile, C is binding to the G again, like that. Okay. So one side unwinding process is going on, and one side in the back side of the RNA polymerase, the rewinding process also going on. Rewinding of the DNA. You can see there's a rewinding process. This is a rewinding process. This is a rewinding process, and this is the unwinding process. Okay. So that's why when the RNA molecule is released, the rewinding of the DNA also takes place. So this is the concept. Okay. Now see the most important part in the elongation part. Okay. So in the elongation part, you can see after the synthesis of 8 nucleotide. Okay. After the synthesis of the 8 nucleotide, jetia 8 nucleotide synthesis hoi jabo. Okay. First they binding with T to A, then they bind G, suppose G to C, then suppose C to G, then suppose T to U, like that. Jetia 8 nucleotide binding hoi jabo. When the 8 nucleotides are completely binded, then the sigma factor is detached from the RNA polymerase. Then the sigma factor is detached from the RNA polymerase. Remember, when the 8 nucleotides are formed during the transcription process, when the 8 nucleotides molecules are formed, after the formation of the 8 nucleotide, the sigma factor is released from the RNA polymerase. Suppose this is the sigma factor, suppose, and this sigma factor is released from this particular RNA polymerase. Okay. So when the sigma factor is released, still the RNA polymerase can synthesize the RNA from the whole DNA process. They can synthesize. Okay. That means in the absence of sigma factor also, RNA polymerase can synthesize the RNA in the absence of sigma factor also. But the initiation process of the RNA polymerase always started with the help of the sigma factor. If there is no sigma factor during the initiation process, then RNA polymerase cannot synthesize the RNA. They cannot synthesize. If in the initiation process, sigma factor is not there. But after the synthesis of 8 nucleotide, the sigma factor is detached from the RNA polymerase. After the detachment of the sigma factor from the RNA polymerase, still RNA polymerase can synthesize the RNA strand continuously, because in that term they do not need the sigma factor. That means what is the function of the sigma factor? The function of the sigma factor is that to help in the initiation process of the transcription. This is the function of the sigma factor. This does start the initiation process. After the starting of the initiation process, they are detached from the RNA polymerase. Okay. So this is called elongation process of the transcription. Then important point. Per second, in every second, RNA polymerase can add 40 bases. This is the speed of the RNA polymerase. 40 bases can be added in the DNA per second. This is the speed of the RNA polymerase. They can synthesize up to 40 bases per second. Okay. So this is called as the elongation process of the RNA transcription in case of the prokaryotes. Okay, just to remember, in the prokaryotes, they having the DNA dependent RNA polymerase. The initiation process start with the sigma factor, and after the formation of 8 nucleotide, the sigma factor is detached from the RNA polymerase, but still the RNA polymerase continued the transcription process. One side they transcribe the RNA synthesis, and another side the DNA again rewinding. One side unwinding occurs and one side rewinding of the DNA occurs. Okay, and just to remember, the template strand always have to be the 3' to the 5' direction. The template is 3' to the 5' direction, and the synthesis always started from 5' to the 3'. You can see the template. This is the 3', this one. So this is the template, 3' to the 5'. You can see 3' to the 5' direction. This is the template. Okay, 3 to 5, and synthesis started this direction. This is this direction of synthesis. That means this is the 5' to the 3' direction. This is the direction. Okay, So direction of synthesis of RNA is 5' to the 3', and the template always have 3 to 5. Okay. So this is the elongation process of the transcription occurring in the prokaryotes. Okay. Then the last one, termination. How the termination occurs in the transcription process of the prokaryotes. How the termination process, how the transcription process is stopped. Put stop hobo. It is okay. So this is the termination process. Okay. So in the termination process of the transcription of the prokaryotic transcription process, you just remember one specific region is there. One specific region is there in the DNA. So DNA having a specific region, and this region is called inverted repeat base sequence. Remember, inverted repeat base sequence is found in the DNA. Okay, and this sequence looks like this. This is the sequence of the termination sequence. This is called the termination sequence. You can see. Suppose the first sequence is A, B, C, D, E, F. Then in between a new sequence is there, and the opposite you can see F, E, D, C, B, A. This is called inverted. This is the inverted sequence, and this is the another sequence. The whole sequence is called the termination sequence. This is the whole termination sequence. Okay, so you can see this is the diagram. This is the diagram of the termination sequence. You can see. You can see A, G, C, T, C. So this is one sequence. You can see this sequence is opposite over here, inverted. You can see in the presence of A over here you can see T, because T can bind to the AAA. Then over here ZZ, over here you can see CC. Then over here C, over here Z is there. Over T is there, over A is there. And over here CC is there, over here ZZ is there. That means this is the opposite sequence. Opposite sequence of this sequence. So this type of sequence is called inverted sequence. And in between a new sequence is there, which is not inverted. These are not inverted sequence. You can see a small sequence is there which is not inverted. Just like XYZ over here, TTT is there. Okay. So in the termination sequence, this type of sequences are there. When this type of sequences are found, then over here the termination of the DNA transcription occurs. That means the RNA polymerase stop transcribing the RNA when this type of sequence is found in the DNA. Okay, and this type of sequence is called termination sequence, or maybe called as an inverted sequence. Inverted sequence. Okay. Now see.
Sir, mane jetya sequence to pai jay tetya mane stop ho jay no.
Yes.
Transcript.
Yes. When this type of
sequence. Sir, RNA polymerase to mane recognize korila je etya mane aru he ko nori mane.
Yes.
Transcribe ko nori. Etu jetya recognize korila.
Yes, absolutely correct. When the RNA polymerase reaches this type of sequences, then what happens? RNA polymerase recognize that we have to stop the transcription process. They can sense when they reaches this type of sequences. Okay, and this sequence is called termination sequence. Okay, so remember in the termination sequence, both the sequences are inverted. You can see A, B, C, D, E, F, and this is the exact opposite sequence is found over here. Exact opposite sequence. Over here you can see binding over here. You can see that's why it is the opposite sequence. In terms of AAA over here, TTT is there. For ZZ over here, CC is there. For C over Z is there. For over T over is A is there. And for CC over is ZZ is there. Like that. So this is called inverted sequence. Okay. And in between both the inverted sequence, a new sequence is there which is not inverted. And when the RNA polymerase senses this type of sequences, then immediately they stop the RNA transcription process. Now see, in this sequence, two other sequences are there which is also very important. Okay, you can see this is a region. This is a region of the inverted sequence. This region is called high GC region. High GC region is called. Now what is high GC region? So in this region, the content of G and the C is very high. You can see GG, CC, GC, GC, GC is like that. So this region is called high GC region. Then this another region is there. This region is called high AT region. High AT region. You can see AT is here. High AT region. Okay. And this is the last sequence AT. This is the last sequence AT where the transcription is stopped. You can see last base transcribed. So this is the last base which is transcribed at the end. So RNA polymerase is moving like that, and when they reaches this base pair, when they reaches over here, the RNA polymerase is detached. When the RNA polymerase is detached from this region, then RNA transcription is completely stopped. Okay, so this sequence is called termination sequence. So in the termination sequence, different regions are there. One region is called inverted sequence, one region is called high GC region, and one region is called high AT region. When they reaches the high AT region, the last base, at the last base of the high AT region, the RNA polymerase stopped and they detach from the DNA. When the RNA polymerase detached from the DNA, then immediately the transcription process is stopped and the RNA strand is released from here. Over here, the RNA strand is completely released from this point, this detached, and the RNA strand is released. Okay,